Every living cell in every living organism carries the substance which controls and dictates how that organism will develop and the characteristics of its offspring. The substance is deoxyribonucleic acid, DNA. How does it function? DNA is extremely complex. This model shows only part of the long chain of the many thousands of atoms comprising a given DNA molecule. Here's a simplified, diagrammatic version of part of the DNA chain. It still looks pretty complicated, so let's see how it's built up. There are two main strands, wound round each other in the form of a double helix. Each strand is made up of phosphate groups, here coloured yellow, and deoxyribose groups, coloured green. The two strands are linked together by basic groups which are attached to the deoxyriboses. The bases on one strand are joined to those on the other by hydrogen bonds. 4 different bases are involved. Cytosine, which can only link with guanine, and thymine, which can only link with adenine. The bases can only pair in this way, cytosine with guanine and thymine with adenine. You can see that here, C always linked with G, A with T. So, DNA consists of long-chain molecules in which 2 spiraling strands are linked by pairs of bases in the way we've just shown. The bases may occur in different orders in different DNA molecules. Now, an important feature of DNA is that it can replicate itself, make copies of itself. This is how it does it. The 2 strands come apart. Each separate strand now builds up a new strand alongside. The new strand is built up from separate nucleotides, each a combination of a phosphate group and a deoxyribose group to which one of the 4 bases is attached. The base finds its complementary base on the DNA strand. Here the base is thymine, which links up with adenine. Here, cytosine links up with guanine. And so on, until a DNA double helix has been formed once more. The same thing happens to the other strand from the original DNA molecule, so that now we have 2 DNA molecules. This is how a given DNA molecule can make any number of copies of itself. How does DNA carry genetic information so that a particular plant or animal develops in the way it does? Slightly simplified, it works like this. The spiral unzips again, and this time a slightly different strand builds up alongside the DNA strand. This is ribonucleic acid, RNA, in a form called messenger RNA. It's like DNA, except that the green deoxyribose groups are replaced by ribose groups, here coloured orange, and at the same time the base thymine present in DNA is replaced by uracil, here coloured purple. The sequence of bases along the RNA strand matches the sequence of bases along the DNA. This messenger RNA, and remember this is only a short portion of a very long chain, now moves out from the cell nucleus where the DNA is to the ribosomes in the cell. Here, proteins are built up. This is how the process works. Molecules of transfer RNA, each with groups of 3 of the 4 bases, attach themselves. Each carries a particular amino acid, depending on the sequence of the 3 bases. This one, with uracil, adenine, and cytosine, carries an amino acid represented by the grey rectangle. It finds its place by base pairing on the chain of messenger RNA. A chain of amino acids is built up, each amino acid corresponding to a particular triplet of bases or anticodon on the transfer RNA, which fit the corresponding triplet or codon on the messenger RNA. The genetic code is a triplet code controlling which amino acid comes at each point in the chain. The sequence of amino acids in the chain has been determined by the sequence of bases in the original DNA of the cell nucleus. The amino acid chain now peels off from the RNA. It's a polypeptide, part of a much longer molecule, which is a protein. What particular protein it is has been determined by the sequences of bases in the original DNA. A gene is any portion of the DNA chain which affects a significant part of the primary sequence of a particular protein. Genes regulate the synthesis of proteins, and the proteins, structural and enzymatic, determine the characteristics of any living organism. So DNA, which controls protein production, therefore determines the characteristics of the plant or animal in which it's present. Now, any change in the DNA carried by an organism will mean that a gene is changed. As a result, a different protein will be produced on the new code carried by that gene. Suppose, for example, that due to chance exposure to radiation or to some mutagenic chemical agent, a thymine group is lost and replaced by cytosine. This is the messenger RNA chain which the DNA programmed before the change. But it no longer fits because cytosine doesn't link with adenine. The new messenger RNA will have no adenine but guanine in this position. In the cell ribosomes, this altered messenger RNA will now build up a slightly different protein. The transfer RNA with the anticodon guanine, uracil and adenine, carrying an amino acid represented by the pink rectangle, no longer fits at this point because uracil will not link with guanine. A different unit of transfer RNA with the anticodon guanine, cytosine, adenine does fit, but of course it carries a different amino acid, the green rectangle. The polypeptide now carries a new amino acid at one point. We have, in fact, a changed primary sequence in the protein. A mutation, a change in the DNA, in the gene, and certain changes cause much more drastic effects than the example we've taken, leads to a change in the protein a gene produces, and a change, useful, harmful, or maybe neutral, in the organism's characteristics. Unpredictable changes in DNA are important contributors to the genetic variability we find in all living things. Genetic variability, then, can be caused by unpredictable mutations, changes in genes, which are inherited by future generations. The giraffe didn't get its long neck through stretching high for young foliage and passing this on as an acquired characteristic. Natural selection of individuals which happen to have necks at least slightly longer caused this particular species to evolve. In the same way, the copper-tolerant grasses in another of these films were the progeny of individual plants which happened to possess copper tolerance before copper salts ever polluted the soil. They were selected by the polluted environment. An important experiment, first carried out by the Ladybirds, demonstrates how we can obtain a population of living organisms specially adapted to a certain environment which can't have acquired its adaptation through exposure to that environment. At Newcastle University Department of Genetics, a version of this experiment. The flask contains a nutrient broth in which the bacterium Escherichia coli is growing. A little of the broth is run onto a nutrient agar in this dish. The spreader is flamed to destroy any unwanted bacteria contaminating it. As with all microbiological procedures, strict precautions must be taken. So this is a normal population of Escherichia coli on a nutrient gel. It's incubated for 24 hours at 37 degrees centigrade. Now for the replica plating technique. This is the Lederberg's replica plating experiment. A piece of sterile velvet is stretched tight, and the gel carrying the Escherichia coli colonies is pressed firmly down onto it so that some bacteria get transferred to the velvet. Now these two dishes contain nutrient agar gel to which an antibiotic, streptomycin, has been added. Escherichia coli should be destroyed by streptomycin, so shouldn't grow on these gels. Each is pressed in turn onto the velvet, which carries traces of the Escherichia coli colonies grown on the normal agar. These plates, with the antibiotic present, are now incubated. Let's look at the result. The two plates containing antibiotic are at the bottom. The original plate without antibiotic is at the top. The original master plate has many hundreds of colonies of Escherichia coli. The two antibiotic plates each show just a few colonies. These are in identical positions on the two plates, which is why we have to show that the result's repeatable. Each of these colonies consists of Escherichia coli, which must be resistant to the streptomycin, and must have grown from individuals in the same area of the master plate which happen to be streptomycin resistant. So here, for example, on the master plate, which contains no antibiotic, remember, there must be some resistant bacilli. We check that the two replica plates are indeed identical, as they should be. The vertical lines serve as guidelines. Then we compare the master plate and mark on it one of the positions from which the replica plates picked up some of the streptomycin-resistant Escherichia coli. A ring is punched around the nutrient gel in the marked area. Within the circle, at least some Escherichia coli must be streptomycin-resistant. A spatula is dipped in alcohol and flamed. It must itself be sterile, and the disc is removed. It's placed in a flask of nutrient broth. In the incubator, the Escherichia coli, including resistant individuals, will multiply. Here's the culture. It's diluted 10 times so that the bacilli are thinned out a little. Then, again taking all sterile precautions, it's spread on a fresh plate of normal nutrient agar. This new master plate is then incubated. Once again, using a piece of sterile velvet, two replica plates are made from this master plate. The replica plates contain, as before, nutrient gel plus streptomycin. Only resistant Escherichia coli transferred from the master plate will grow on these two replica plates. The result: master plate at the top, replica plates containing antibiotic below. We can see that there are more colonies of resistant Escherichia coli than there were the first time we did this, as we should expect, since we deliberately tried to select resistant bacteria from a portion of the original master plate. Once again, the replica plates are used to find an area on the master plate where there are streptomycin-resistant bacteria. Gel is taken from this area, the culture diluted again 10 times, and the whole procedure repeated. From our third master plate, again at the top, we get two replica plates containing an even larger number of streptomycin-resistant colonies. After 6 stages, we reach this. The master plate at the top precisely matches the 2 replica plates at the bottom. Bacteria picked up on the velvet from each of the colonies at the top have grown successfully on the replica plates in the presence of streptomycin. All the Escherichia coli on the master plate must be streptomycin resistant. We've achieved a completely antibiotic-resistant population, although the bacteria on this top plate have never been exposed to the streptomycin. The resistance cannot have been acquired, therefore, by exposure to the antibiotic. A resistant population has come about by a process of selection of already resistant individuals. We can't prove that the theory of evolution is true, but it remains the most satisfactory interpretation we have of the kinds of evidence which we've looked at in these films. We started with the evidence from the past, the fossil record. Although there are many gaps, fossils give us a broad picture of how plants and animals developed throughout the thousands of millions of years of the Earth's history. And some fossil sequences provide a quite detailed record of how certain living things have evolved through time. The homologies shown by living things provide evidence of a different kind. Again, we illustrated only some, the structural homologies between the forelimbs of very different animals and between insect appendages. Some behavioral homologies amongst, for example, waterfowl. And most recently investigated the remarkable biochemical homologies revealed by the study of proteins. These homologies and many others suggest common ancestries for different groups of living things, an essential feature of evolutionary theory. We looked at ways in which isolation has acted upon small founder colonies, like the rodents on the Orkneys and Shetlands, and saw how these reveal evolution, change, going on today. And we looked at other examples of selection in action, using experimental studies of banded snails, of metal-tolerant grasses, which provide a very vivid illustration of how an environment selects particular varieties, and of the all-important cereal crops, wheat and barley. These were just some examples of natural and artificial selection which help us to understand how evolution works. Finally, we looked at the way in which vital genetic information is encoded by DNA in the cells of all living things, whether dandelions, people, or fruit flies. We now know that changes in DNA produce the unpredictable inherited variations on which selection can then act. Selection, acting on an enormous range of genetic variability, has led to the tremendous variety of living things on Earth today, including ourselves. This view of the mechanism of evolution provides the best explanation we have of the fascinating variety of life in the world around us, in the past, present, and as far as we can know, in the future.